research is quite similar to Laura's. I look at the impacts of land use on hydrology and water quality, and more specifically, I look at the occurrence, fate, and effects of emerging contaminants in the environment. So in addition to my research, I'm also program manager for our MSc in River Basin Management, and I'm also Associate Director of Water at Leeds. So it's my pleasure to welcome you to this virtual seminar of the Global Food and Environment Institute, Emerging Contaminants in the Agro-System, which is going to be presented by Dr. Laura Carter, who is Associate Professor in Soil and Environmental Chemistry here at the University of Leeds. So Dr. Carter's research focuses on understanding the fate and uptake of emerging contaminants in the natural environment, with a particular focus on soil and plant systems. She currently holds a prestigious UKRI Future Leaders Fellowship that funds her project Contaminants of Emerging Concern in Agricultural Systems, a Risk to Soil and Plant Health. So this project's using multidisciplinary approaches to evaluate the global risk of chemicals in agricultural systems. A little bit about the Global Food and Environment Institute. So this is an interdisciplinary research community which brings together members from across academia, industry, and public policy to work on the integrated challenges of food security and climate change. So if interested, you can get further information from the GFEI website. And this seminar is also part of a wider series of seminars, so you can check the chat function with links to that series on Eventbrite, and also the GFEI website and our Twitter feed. Just like to remind you of seminar etiquette. So if you can ensure that your cameras and your microphones are switched off for the duration of the seminar. The presentation by Laura is going to take about 20 minutes. And during that time, please do feel free to use the chat function to put in any questions that you have and make sure that they're addressed to everybody. And then at the end of Laura's presentation, there'll be a 10-minute Q&A session where I'll collate some of those questions and put them to Laura. Can I also remind you that the seminar is being recorded, and that recording will be made available via YouTube. So over to you, Laura. Thank you.
Thanks, Paul, for the introduction, and to the Global Food and Environment Institute for the invitation to come here today and present to you some of the work which I'm undertaking at the moment at the University of Leeds. So I guess just a little introduction. As Paul said, my interest is around emerging contaminants in the environment, and today I'm going to be talking specifically about pharmaceuticals in agro-ecosystems. And I started working in this area as a PhD student over 10 years ago. And as you can see from this picture of me looking very keen, I was about to go in the field and do some sampling. There's probably a little less sampling and more desk-based work at the moment, but hopefully today I'm going to give you a snapshot of some of the exciting work which we're currently undertaking. So I've tried to divide the talk into three main parts. So first of all, focusing on the pathways and how pharmaceutical contaminants can enter the environment, and then move on to think about the fate and the behavior of these contaminants in the environment. So what happens to them? And then finally, try and bring this all together to think about what this contamination means in terms of ecosystem and human health risks. So to start with, the pathways. So pharmaceuticals are ubiquitous contaminants. They are found all over the globe in our soils, our sediments, our lakes, our rivers. And thinking about human pharmaceuticals in particular, the reason why they're ubiquitous in our environment is that following their use, a portion of the active ingredient is released into the sewage. And wastewater treatment plants are not specifically designed to remove these chemicals. So they are released into the environment in the wastewater byproducts, including effluents and sludges. And what's important to remember here is that these contaminants, so pharmaceuticals, have retained biological potency. So when they're given to a human for a therapeutic purpose, they're designed to elicit an effect. And it's this potency which isn't lost in the environment when these chemicals are released. And this is where my interest is. So thinking about the soil environment in particular, the two main pathways which I'm going to focus on for the rest of this talk include the use of sludges or biosolids as a soil amendment, and the use of wastewater effluent or reclaimed wastewater as a source of irrigation. And these are commonly used in agricultural systems and present one of the main pathways by which these chemicals can enter and become present in agricultural systems. But it's also important to think about veterinary medicines as well. So taking antibiotics as an example. So when these are administered to livestock, they are also excreted directly onto land and also have this retained biological potency. Now, irrigation with wastewater is a widely carried out practice in countries which typically suffer from limited freshwater reserves. So examples would include countries in Southern Europe, as well as Mexico, where wastewater irrigation has been practiced for over 100 years. And then Israel as well. So this picture was taken a couple of weeks ago when I was visiting Israel. And they use about 80 to 90% of their wastewater for irrigation on agricultural land. And following wastewater irrigation, we know that a range of different pharmaceuticals can be detected in soils. So I've just included some examples here. So commonly detected pharmaceuticals include antiepileptics, antibiotics, you've got painkillers, and these are typically in the range of around microgram per gram to nanogram per gram concentrations. Now in the UK, we do not use wastewater as a source of irrigation, but we do use biosolids, slurries and manures as a soil amendment, so for agricultural fertilizer reasons. And at the moment, we are lacking a baseline data set of what soil contaminants are present following the use of these amendments. And this is something which we're trying to address with our project SPADE. So we're in the process of analysing soils which have been collected from farms all across the UK. And these soils have been in receipt of either manure, slurry or biosolids. So we're analysing these soils using two different approaches. So we're carrying out a targeted analysis. So we have identified a list of chemicals that we think are going to be present. And there's known contaminants in biosolids and manures. And then we're going to do some analysis to try and work out whether they're present and the concentrations of these chemicals that are present. But then to complement this, we're also doing a non-targeted analysis, which is going to create this effectively a chemical fingerprint of the sample. So it's not specific to working out concentrations of a particular pharmaceutical. It's broader than that, and it's going to really show us some idea of what is present in these soils following the use of these amendments. So as I said, the analysis is underway, and so we're hoping for some results in early 2023. And hopefully that's provided a bit of context as to how pharmaceuticals can enter and become present into the environment. And then this moves me on to talk about fate and the behavior of these chemicals. So what I'm trying to get across here in this slide is that chemical exposure in the environment, or specifically soil plant systems, is really complex. And I've tried to summarize some of the main processes which are important when you're thinking about the fate and the behavior of pharmaceuticals. But it's important to bear in mind that these processes are dependent on the environmental conditions. So thinking about soil type, the prevailing climate, for example, in combination with the physicochemical properties of the pharmaceutical itself. So some examples here are mechanisms by which pharmaceuticals can be removed from the soil plant systems. And these could include, for example, photodegradation or microbial degradation. And so this would reduce the concentration of the pharmaceutical in this system. We also know that pharmaceuticals can move throughout soil plant systems. So in the soil, processes could include, for example, leaching or runoff. And what's really key here to controlling the mobility of these chemicals is their potential to sorb or desorb to the soil. And I think the key take-home message from this slide is that when a pharmaceutical accumulates and can undergo a number of different processes which can change where the chemical is and the amount of chemical that's present. And this is ultimately going to be a key factor in understanding what the risk is from pharmaceuticals. And I'd just like to highlight one of the latest pieces of work, which is looking at the fate of antimicrobials. And this is some work which was led by a PhD student that Paul and I supervised, looking at how differences in manure application technique can affect the leaching behavior of antibiotics. So leaching occurs when you have excess water and it removes the pharmaceuticals from the soil, for example, through surface runoff or drainage through the soil profile, for example. And from John's work, he found that common types of manure application result in differences in the loss of antibiotics from soils. So techniques that involve more incorporation methods, such as shallow disc injection, for example, reduce the loss of antibiotics from soils from surface runoff and leaching, in comparison to when the manures were applied via surface application, so when they were spread directly onto the soil. Now, plant uptake is also another key process when thinking about the behavior of these chemicals in the environment. And this is the focus where I'm going to really sort of continue the rest of the presentation on. So it's looking at the work which we're doing at the moment on the plant uptake, but also the effects of pharmaceuticals in soil plant systems. So going back a few years now, I guess some of my earlier work was able to demonstrate that plants can take up and they can accumulate pharmaceuticals that are present in soils. And what we were also able to show was that pharmaceuticals could be metabolized by the plants. So here in this slide, I've got an example of a benzodiazepine study which I carried out. And we have diazepam here. So this is prescribed for the treatment of anxiety. And it was added to the soil. And then I analyzed the plant material for residues of this compound, but also any potential metabolites. And what we were able to see in this study is that we could detect diazepam, so the parent compound, but we were also able to detect the known metabolites of diazepam. And what's interesting with benzodiazepines is that temazepam and oxazepam, for example, are actually prescribed in their own right for the treatment of anxiety. So when we're talking about the risk coming from the biological potency of the pharmaceuticals, when they're metabolized, this doesn't necessarily get rid of that risk, because the metabolites themselves have this or can have this retained biological potency. And I guess giving this the chemical potency of the pharmaceuticals, it probably comes as no surprise that following the uptake and accumulation of pharmaceuticals into plants, these chemicals can induce toxic responses. So this slide summarizes a couple of studies which we've done, which have looked at toxic responses in plants following exposure to pharmaceuticals. However, what I'd like to point out here is that most of these studies have been carried out at very high concentrations. So here we're talking about milligram per kilogram concentrations. And if you can think back to the slide where I was talking about the types of chemicals that are detected in our soils, these are typically at nanogram per gram concentrations. So this has then led on to some of my current work, which is part of my Future Leaders Fellowship, which is looking at what's happening to the plants at a sublethal level. So yes, we've worked out that at high concentrations, these chemicals can be toxic and can induce lethal effects in the plants, but what's happening at a lower environmentally relevant concentration, and are any of the important processes in plant growth and development being affected by the presence of these chemicals? So this is where the work on mycorrhiza fungi has come into this, because following circularity principles and a sustainable approach to agriculture, we apply and we use soil amendments such as biosolids and manure, but we also are including the use of arbuscular mycorrhiza fungi, or AMF for short. And these are because they're biofertilizers, and they can increase soil structure, decrease erosion. And these are just a couple of examples, but they are advocated under regenerative and sustainable agricultural processes. Now, almost all plants are engaged in a symbiotic relationship with mycorrhiza fungi. So the soil fungi promote and support plant growth because they supply nutrients to the plant roots in return for carbon, which is fixed by photosynthesis. So the fungi are attached to the roots and basically elongate the reach to the nutrients and the water, which are then taken up into the plant, and in return, carbon is sent down to the roots. So we know that the relationships, these symbiotic fungal partnerships, are really important. But at the same time, we know that antifungals have been detected in biosolids that are being applied to agricultural lands. So what this experiment, or what this series of experiments set out to do, was to understand what this means for plant health and the important role that fungi play in delivering the minerals to the plant. So this study is in collaboration with the University of Sheffield and the University of York, and we used radio and stable isotope tracing techniques to track the movement in the soil, from the soil into the plant for nutrients. So as you can see in these pictures, these are some of the plants that were grown in the wheat exposure, which we carried out. And we have these PVC cores in the pot, and these have windows cut out of them in this fine mesh, which is covering the windows. But the mesh, the fungi can go through the mesh and can access the core contents. So what we did to differentiate between the fungi that were taken up into the plants that were driven by the fungal relationships in comparison to the passive diffusion of the nutrients, we rotated one of the cores in each pot, and this severed any fungal connections between the plant roots and the core contents, whilst the other core remained static. So taking the wheat study as an example, we added radio-labeled solution of nutrients to each of the cores, and then we were able to see the accumulation between the static core and the rotated core as to whether this was being driven by fungal networks or not. So we were able to calculate the fungal acquired nutrients by subtracting the content of the phosphorus detected in the plant tissues where the isotope tracer was added to the rotated core, and the amount that was detected in the plants where it was added to the static core. And this is where I'd like to bring in the results. So this is the first set of results is from the wheat study. So what we were able to show here is that the presence of the antifungal had a negative effect on the movement of phosphorus from the roots into the wheat plants. So the box plot on the right-hand side represents the fungal acquired shoot concentrations of phosphorus. And as you can see here, we had a reduction in the fungal acquired accumulation of phosphorus in the roots following the exposure to the antifungals. But what was really interesting was that we didn't see any impacts on the degree of the colonization of the roots by the fungi. So effectively, in the drugged soil and the no drugged soil, we saw the same amount of fungal colonization. And this led us to suggest that the fungi were not at, the antifungals were not at concentrations at which were lethal to the fungi or inhibitory in their ability to colonize the roots. But what they did hamper was the facilitation and the movement of phosphorus from the soil into the plants. And again, this is just to highlight the same effect, but in a different species. So this is with spring onion, and we saw a reduction in the amount of phosphorus that was able to be transported, moved into the spring onion in the soils that contain the antifungals. But interestingly, in this crop, so in the spring onion, there was an effect on the colonization of the fungi by the roots. So this shows that the effects that we're seeing are crop specific. And I think this poses some really interesting questions from an agricultural management perspective. Okay, so coming towards the end of my talk and the final few slides, I want to move on to think about what this means in terms of risk. So quite often when I tell people what I do and the research area that I'm interested in, the first question people have is, can I still eat my vegetables? Because there's a lot of concern that we are consuming produce that may be contaminated, and people want to know what impact that has on human health. And this is a really, really hard question to answer, but I think the key points here are, if we want to think about the concentration that we're seeing these chemicals accumulate in the crops and compare that to a therapeutic dose. So what you would be prescribed in terms of taking some medication, for example, the concentrations in the plant are very low. So if we take radish, for example, you'd have to consume hundreds of radish to get anywhere near a therapeutic dose of the level of benzodiazepines, for example. But what we have to think about here is long-term exposure and susceptible populations. So thinking about children and the elderly, for example, if we're saying there is little risk to adults, is that protective enough across all people in society? And are there people that may be particularly susceptible to certain types of chemicals in the environment? And I think the other angle we also have to consider in terms of risk is the impacts on agricultural productivity. So we've shown in controlled laboratory trials that these chemicals can impact on plant processes that are responsible for growth and development of the plant. So in a field setting, is this going to have any wider impacts on agricultural productivity? And this is something that we're going to try and assess in follow-up experiments. But lastly, the key point I want to raise here is, when we're thinking about risk, are we actually thinking about the right chemicals? Because quite often the experiments we conduct in the lab are driven by our availability of analytical methods or our interest in a particular compound. And are we actually analysing our plants and our soil and our effect studies? Are they centred on chemicals that are present and are causing the most concern? And so to address this, my postdoc has been working on a framework to assess the accumulation of pharmaceuticals into plants following wastewater irrigation scenarios. And what's different here is that we've taken a step back and not tried to predict a subset of pharmaceuticals. So we haven't defined our own chemicals of interest. We've taken data from a high resolution mass spectrometry scan of influent that's entering a wastewater treatment plant. And then we've used that to calculate concentrations in the effluent, concentrations going into the soil, and then ultimately concentrations that are going to be taken up into the plant. And this has been a very, very complex task to undertake, and I've tried to simplify this as much as possible in this slide. But I'd just like to highlight some of the main take-home messages from what we've discovered so far in terms of this analysis. I guess the key one being that the types of chemicals which we are predicting to be taken up into plants are varied and not necessarily the ones that we're typically working on in research at the moment. So a lot of studies are underway using carbamazepine, acetaminophen, and antibiotic sulfamethoxazole. But they're some of the most highly accumulating chemicals that we predict that are going to be in the plant are chemicals that haven't even been assessed in terms of their risk in soil plant systems. So I think this then raises questions about future research direction and the chemicals that we need to prioritize. So I'm just going to finish now by, I guess, giving a quick summary of what I presented and thinking about the question of risk again. So we know that pharmaceuticals can enter the environment. We know their behavior is variable, but we also know that they can, in certain cases, be taken up and induce responses in plants. But I think it's fair to say that for the widespread contaminants that are in the environment, we still do not know fully the extent to which effects can occur in pharmaceuticals being taken up and accumulated by plants. But what we do know is that it's not necessarily an issue that these chemicals may be present in small concentrations, because we're still seeing effects at small concentrations. And I think this then brings in the wider point that if we are driving to reach net zero and we're using circular economy principles, where we are increasing our use of resources applied to land, so our biosolids and our wastewater, this is only going to increase the presence of these chemicals in the environment. So we need to be making sure that we can operate these practices safely and sustainably. So thank you very much for listening. I'd just like to thank my wider research group and collaborators at the University of Sheffield and York, who have been instrumental in doing this work. And if you'd like to find out any more, then we have a summary animation on YouTube describing the work underway in my group. So thank you, and I'll hand over to Paul.
Okay, thank you, Laura, for a great presentation, fascinating, and for keeping absolutely perfectly to time. So we have 10 minutes now for a Q&A session. So please do add any questions that you have into the chat, and I'll try and pick from those and put them to Laura. So a few questions. Laura, first of all, could you, and I know you touched upon this, could you explain why human drugs are ending up in sludge that's applied to land?
Sorry, you cut out just for a little bit, just to check. You have a question around how are human drugs ending up in sludge that is applied to land?
Yeah.
Okay, perfect. So following use, our bodies only use a small proportion of these drugs, of these chemicals, and the rest is eliminated in our waste products, which then enter the treatment process, the wastewater treatment processes. And the amount that's leaving our body that is unused and unchanged is very variable. So it's dependent on the types of different drugs, it's dependent on the person that's taking the medication. But what we do know is there is this fraction of biologically active chemical with a therapeutic effect. So, or in other words, the sort of the active proportion of the chemical that is being released into the sewage. And then, as I said, the wastewater treatment plants are not designed to remove these chemicals. So some of the treatment processes do reduce the concentration of some of these drugs. Other treatment processes can change and transform the drugs into other substances. But these processes are not removing everything completely. So then they are present in the byproducts that are released into the environment.
Okay, thanks, Laura. Another question. So you talked about the experiment on different methods of applying manures and slurries to fields and the impact that that could have. So which type of slurry application would you recommend that farmers are following to try to limit environmental risk?
I think, well, the results from this study as well were also dependent on the effects of the amendment were different dependent on the type of antibiotic of interest, but the approaches that were ones where the manure was incorporated into the soil, so it was combined, these were the ones that reduced in that were resulting in the least amount of antibiotics in the runoff and in the leachate that was collected.
Okay. Another question is your data about phosphate uptake by plants. So is that due to a direct effect of the pharmaceuticals, or is it some interaction of the pharmaceutical with the phosphorus maybe?
It could be. That's something that we're trying to explore now, because we weren't expecting to see differences between the different crops. So it seems like, I think this is it's a crop specific effect, but I think that it could be revealed. We're doing some microbial analysis to see whether that reveals whether the drugs have had an impact on some of the communities that are responsible for facilitating the uptake, and whether that is species dependent or not is something that we're trying to understand. But it appears that it's also nutrient specific as well. So we have done a similar analysis for nitrogen, and the results are also variable between species. So it's nutrient and it's crop specific. So it's a lot more complex, and we're still trying to unpick it.
Okay, lots of work still in the pipeline there then. So we're covering lots of different areas, working your hard, Laura. So how do you think we should assess the effects of mixtures of pharmaceuticals on human health and on the environment? How do we go about looking at various chemicals that are all present together?
I think there's different ways that you can approach this. So from a usage and exposure perspective, we can use like the modeling framework which we've developed to think about the chemicals that are accumulating in the highest concentrations in the environment and prioritizing those for a mixture assessment. Or we can think about it from a mode of action approach, or if we know that a certain class of chemicals are more toxic or eliciting greater responses in the soil plant system, I think we can target mixture assessment towards those. It's something I think we need to definitely address. So the work which we've done originally started off with doing individual chemical exposures, because it's a lot easier to try and link effects to a single substance in the soil. But in terms of environmentally realistic scenarios, is that what's occurring in the environment? No. So how do we introduce that complexity while still being able to untangle what might be driving the effects? So I think, yeah, there's lots of work to do there.
Okay, and a related question to that, so I guess that was on the risk assessment side of it. There are all these different pharmaceuticals in the soil. To what extent do you think they are interacting? What sort of interactions might be going on?
I think this is a tricky question. I can see you're smiling, Paul. I think this is, if they're present in the environment together, then there's the potential for the interaction, but the behavior is going to be influenced about how they're able to absorb to the soil, for example. So with the presence of one chemical in the soil, does that impact on how the other chemicals are able to absorb, or is it more bioavailable in the pore water? So it's, I don't think we've got the data to answer that question at the moment, but there's going to be interactions in the fate of these chemicals, and then whether there's any preference in terms of uptake, or whether you have the presence of one chemical that can bind to a receptor in a plant and stop another effect happening. I think these are all questions that we need to try and understand.
And so the second part of that question was, are you doing any experiments on that sort of mixture interaction?
We are from, so what I didn't cover this work in my presentation, but we've got a study looking at the phytoremediation of antibiotics in rivers using LEMNA, and mixture assessment and interactions between antibiotics, but also metals and biocides is something that we're trying to unpick. But it's not something that we've got work underway with for terrestrial sort of soil plant systems.
Okay, thanks. Your plant uptake data, so did that come from field observations or lab experiments? And if it was from lab experiments, which I think it was, how repeatable is that data?
So yes, you're right. So the data which I presented today, they were either controlled laboratory exposures in a growth chamber or in a greenhouse. So it's a good question about the repeatability, but I think the fact they are controlled experiments where we have manipulated the treatment or the addition of the chemical to the test system, but we've controlled other factors, would mean that the experiments should be reproducible in that front. In terms of whether they're reliable and an assessment of what's occurring in the natural environment, then it does bring into question around the presence of mixtures. And it's not just a case of mixtures with pharmaceuticals in the environment, you've got whole other complexities to do with other contaminants. And you've got various like climatic factors that could be influencing the exposure. So what we're doing as part of the fellowship is to then look at whether we can see the same effects that we've observed in the lab in the field, to then think about the translation of that work.
Right, thanks. And on the sort of plant theme, so if there are differences between uptake between plant species, should we be encouraging application of these organic amendments to crops which perhaps don't take up pollutants? And I guess that goes, it goes wider than pharmaceuticals.
Yeah, definitely. And I think the method of application as well is important. So thinking about irrigation, you can get, if you've got drip versus the spray irrigation, you can get differences in accumulation between the two different methods. So I think the fact we've observed differences between species, and we also know there's differences in accumulation between methods, means I think you could tailor specific agronomical practices towards less risky scenarios.
Right. And I think this will have to be the final question. There's lots more people wanting to ask you things, Laura, and I'm sure you'd be happy to answer them after this event. But the last one, thinking about the different methods of applying slurry and whether you incorporate it or not, if you're using methods like injection, which perhaps reduce losses in runoff, are you then just increasing concentrations in soil and possibly leading to greater uptake by plants?
Yeah, that's a fair point. I think when it's present in the soil, then we know that there's processes such as microbial degradation, which can then break down these chemicals in the soils, and you're confining the contamination to a particular area where you can then target your mitigation. Whereas if you've got runoff, then it's then sort of increasing the connection between the terrestrial and the aqueous systems, and you're spreading the risk. So I guess it's how do you mitigate, and whether that's easier if you can keep the contaminants in a particular area.
Okay, right. Well, we've run out of time, so I think we'll draw the seminar to a close there. So thanks again, Laura, for a fascinating presentation. Thanks everybody for attending, and I hope you enjoy the rest of your day. Thank you.
Thank you.
Thank you too.
Yeah, thanks very much. Really interesting.
Thank you, Paul, and thank you, Laura, for this wonderful presentation. Thanks, you all.
Thank you all.